So today our next topic is the absorption of nutrients. So if you talk about the nutrients, that was the carbohydrates, protein, as well as fats. So first was the carbohydrate, okay, carbohydrates. And if we talk about the carbohydrates, so there were glucose, fructose, as well as galactose, okay. These were the monosaccharides that were going to be absorbed in the ileum. So protein was there. In protein, the amino acids was there. Okay, the monosaccharide amino acid. After that, we were having a fats. In fats, we were having a glycerol as well as fatty acid. Okay, fatty acid was there. After that, we were having these. These both are called the monoglyceride, as you guys all know. In the fats digestion, we discuss about that. So after that, we were in ileum. As you guys all know, there were the brush border epithelial cells were there. Okay, brush border epithelial cells were there, as you guys all know. Okay, microvilli through which the nutrients got absorbed and moved to the bloodstream and provide us the energy. So if we talk about the glucose, the first is glucose. So glucose is basically get absorbed by the, as you guys all know, in the ileum we are having most of the microvilli, and they increase the surface area of the ileum. Ileum is a third, you can say that, part of the small intestine. So in ileum we were, the absorption mainly occurs. So here we are having the microvilli structure, and which are having a epithelial brush border cells. So through that the absorption basically occurs. So we are, first of all, we are taking a glucose. So glucose basically get absorbed by the active transport. Okay, first of all, let me write all, then I'll be telling you everything. After, if we talk about the fructose, so fructose basically got, you can say that, got absorbed by the facilitated diffusion. Active transport, which means the use of the energy. If you talk about the facilitated diffusion, so facilitated diffusion means that there is any carrier for the diffusion, like for example, any protein is there, any carrier through which the absorption occur, means the you can say that mainly particle will go, not any particle will go, only few particles, selective particle, you can say, that will move through the facilitated diffusion with the carrier. If we talk about galactose, so in galactose, if we talk about, so they are having most of the active transport diffusion, or you can say that sometimes facilitated too, sometimes facilitated diffusion too. Okay, this was the carbohydrate. If we talk about the protein, in which amino acid are the, you can say that these are the monosaccharides and these are the polysaccharides. So polysaccharides cannot absorb because they are macromolecules. If we talk about this, so they are the micromolecules, and that can easily, you can say that, the absorb. So if we talk about the micromolecules, so they can easily absorb, so that's why we have taken here the micromolecules. Okay. If we talk about amino acid, so they also got absorbed by the active transport. Okay, so after that we are having a glycerol. Fats, in fats we are having glycerol fatty acid. Okay, so if we talk about the uh, fatty acid and glycerol, so glycerol is basically a fat soluble that can easily, you can say that, the dissolve or absorb. If we talk about the fatty acids, so they are the hydrophobic. They are hydrophobic, means they cannot, they are not water soluble. So there will be some carrier to absorb the fatty acid. We'll be discussing that. Okay, so if we talk about the glycerol, so glycerol will get diffused by the simple diffusion. Okay, because it can easily move to the cell membrane. Okay, so here we are having some things to be discussed. So first of all, let's talk about the glucose. Let's talk about the glucose. So if we talk about the glucose, as you guys all know, I told you guys, will be absorbed by the active transport. Let's see how. Okay, active transport of glucose. Active transport of glucose. Okay, active transport of glucose. So here is the where energy is to be used. Energy is to be used by protein chain or anything. Okay, so here we are having a pump, sodium potassium pump. This is called the, let me tell you, this is called the sodium potassium pump, through which the active transport basically occur of the glucose. Active transport, what does it mean? It means that where the energy is be used. So here the 1 ATP will use. Okay, 1 ATP will use, and through the sodium potassium pump, the 3 sodium ions will move outside, as well as, and the, you can say that 2 potassium ions, sorry, so 2 potassium ions will move outside. Okay, so here you can see that sodium concentration is more, and then glucose will attach with that the sodium, and that will be moved into the, through the ATP, which is 1 ATP. So through this sodium potassium pump, the glucose get easily absorbed. Okay, because of the high concentration of the sodium ions. Okay, sodium ions, and sodium ions will get attached with the glucose, will get attached with sodium ions, and it will easily get absorbed. After that, we are having another molecule, that was fructose. Okay, if we talk about the fructose, this is also a monosaccharide. So if we talk about the fructose, so for example, we are having a cell membrane biphospholipid layer. This is the, they are the biphospholipid layer of the cell membrane. Okay, of the cell membrane, these are the brush border epithelial, through which the fructose will move into the bloodstream, and after that blood vessels to the blood, into the blood. So let me make, so here you can see that these are these, this is basically a this is the this is a molecule, this is the protein, this is the extrinsic factor through which the fructose will get attached, like this. Fructose will get attached, and that will move inside the cell membrane. Okay, so as you guys all know, I told you guys that fructose will move through the facilitated diffusion. So what is facilitated diffusion? Here you can see that facilitated diffusion is a diffusion where the absorption, you can say, through where there is any carrier to take the molecule and to get absorbed. So you can see that here we are having a carrier, this is a channelized, this is a protein. Okay, so here you can see this is a carrier through which the fructose is moving inside the bilipid layer of the cell membrane. So here you can see that this, there is any carrier for the, for taking the molecule and move inside the cell membrane. So that is called the facilitated diffusion, that selective particles or selective molecules are moving inside the cell membrane with the help of the carrier. So here you can see that we are having a carrier, that is extrinsic factor. So this is a protein, and this is basically facilitated a protein, which means helping protein, and this is the channelized protein outside of the cell membrane. Okay, after that fructose will move inside the cell, and protein will get detached. This extrinsic fructose will be there in the cell membrane, then this protein will get detached from the fructose, and that will move outside at the same place. So here you can see that the fructose is now in the cell membrane, so that can easily absorb. Okay, so this was the absorption of the fructose. Now if we talk about the galactose, so galactose will be also same as the this one, active transport, there is no need to tell. So after that, amino acid is also like the active transport, here you can see that. Okay, so after that, the main thing was the glycerol. So as you guys all know, glycerol is a fat soluble. It can easily absorbed by the simple diffusion. What is simple diffusion? Simple diffusion means that the absorption through which the particle get easily diffused or easily absorbed. Not selective particle is there, like here the protein carrier. So there is no carrier that can easily absorb. So there is no, and I need to tell the glycerol will easily absorb in the cell membrane. Okay, after that we are having a main thing, that is fatty acid, because fatty acid is a hydrophobic. How it will get absorbed, let me tell. So we are having a next, that is the fatty acid. So if we talk about the fatty acid, so fatty acid is the monoglycerol. As you guys all know, I told you guys about that in the digestion of the fats. So this is what, this is a hydrophobic, which means that it cannot easily diffuse in the cell membrane. Hydrophobic. Okay. After that we are having, here you can see that this is the molecule of the fatty acid. Okay. So bile salts will get, this is the molecule of the fatty acid, and here you can see the bile salts will get attached with the fatty acid. This bile salts will be doing the absorption of the, with the help of the bile salt, the absorption of the fatty acid will get occur. After that we are having a bile salts. In bile salts, here you can see that in bile salts, here you can see that this is a hydrophilic. Okay, hydrophilic, which means that it can easily absorb, and this is the hydrophobic, that is attached. Okay, so this bile salts will cover the fatty acid. Okay, after that the pinocytosis will occur. What is pinocytosis? Pinocytosis means the water engulfs. And so as you guys all know, the bile salts has engulfed this bile fatty acid. Okay, after that it will after that we are having like this is the bile salts. And here you can see that fatty acid molecule is there. So this is what, this is the foldings, like here you can see that this is the foldings. Okay, here the fatty acid there, and these are foldings. So these foldings of the cell membrane is called the endocytosis. Okay. So through this the fatty acid will move inside the, here we were having epithelial cells, as you guys all know. So fatty acid will move inside with bile salts, and after that moving of the fatty acid inside the cell membrane, the bile salts will get detached. So here you can see that we are having a fatty acid molecule, and bile salts will get detached. So with the help of the bile salts, basically the absorption of the fatty acid basically occur. So bile salt will cover the fatty acid. One process I told you guys, that is bile salts. So bile salt basically cover the fatty acid. Fatty acid, after that the pinocytosis is occurring, as you guys all know, as you guys I told you guys. After that we are having endocytosis, like this endocytosis. Okay, in endocytosis process the bile will detach with the fatty acid, and the fatty acid now is in the cell membrane. After that the covering of the in the fatty acid which is a cell membrane. So the covering in the cell membrane, so the covering will occur to the fatty acid by with the help of the protein, and that will done by the Golgi bodies. Golgi body, the Golgi bodies is basically a we are called that as a post cell of the cell membrane. So we are having a Golgi bodies. So what is Golgi bodies? Golgi bodies is basically a organelle which did the which do the which form the vesicle outside of the any molecule. Okay, so after Golgi bodies, in you can say that the inside cell, after the upper side of the Golgi bodies we are having the you can say the endoreticulum, or you can say that the rough endoplasm reticulum. Okay, is present. So protein will come by the this ER, and after that that will move to the Golgi bodies, and Golgi bodies will having that will do the covering of the protein, and that protein covering is called the modification. Okay, modification that is called, for example, like that. This is the this is was the molecule, and there is the covering. Okay, this covering is done by the Golgi bodies, because the work of the Golgi bodies is to make the vesicles surround any kind of a molecule. So here you can see that the vesicle is produced. So that particle we are calling that as a chylomicron. Chylomicron. This is in white in color, and this is made up of the lipoprotein. Lipoprotein. Okay, so that chylomicron will basically move to the submucosa, where the lacteal is there. We are having a submucosa. Okay, in submucosa we are having, here you can see that we were having a cell like this, epithelial cells were there. Okay, and here we are having a lacteal. A lacteal is there. Lacteal is what? Lacteal is basically a lymphatic vessel which is in white in color. Okay, so so that which means that it will move into by with the help of the lacteal, and after that chylomicron will move into the lacteal, and that will move into the blood, and the absorption will occur through the lymphatic system into the blood. Okay, so so as you guys all know, we were having a chylomicron. So what is chylomicron? As you guys all know, I told you guys, chylomicron is basically made up of a lipoprotein, and that is having a low density, lipid. Now it can easily absorb into the blood. Okay, so there are two ways of the absorption of the chylomicron. So first one is the hepatic portal vein. Hepatic portal vein. Hepatic portal vein, which is coming from the liver. So that will through that the chylomicron will get attached, and after that will move into the liver for the storage or in the modification. Okay, after that we are having a second, that is the chylomicron will move through the lymphatic vessel lymphatic vessel into the thoracic lymphatic vessel lymphatic vessel. Okay, and after that it will move into the subclavicle duct. Okay, so when the chylomicron will move into the blood vessel, and then it got break, and after that it will help into the respiration. In the respiration we are having a, and you can see that in the respiration, and it will store into the adipose tissue, which are basically in our skin, store in adipose tissue. Okay, that will store in the skin. So if it is moving into the liver, then it will break by the lipase. As you guys all know, the it will break into the lipase by the lipase. Lipase is the enzyme through which the basically a fatty acid, and when these uh, things basically got break, so through that easily it can easily get absorbed. So here we are having a fatty acid. If we talk about the glycerol, as you guys all know, glycerol can easily absorbed by the epithelial cell, but fatty acid will join with the bile salts, and after that making a chylomicron, and that after that it will move into the bloodstream. So let me give you the so here the small intestine is getting ended, so let me give you the overview. So first part we were having a duodenum. Second part we were having a jejunum of the small intestine. After that we were having a ileum. So if we talk about the jejunum, here the jejunum, the not jejunum, here duodenum is about 3.5 3.5 meter. Okay, duodenum. So this here the only the protein get digest. There is a complete digestion of the protein. If we talk about the jejunum, so jejunum is basically a you can say that there is 2.4 meter, and if we talk about the, so this is the uh, two half part of the small intestine. Okay, so jejunum is covered by the two half point part. Okay, after that, if we talk about the ileum, so ileum is basically the third fifth part through where the helium is covered. So if we talk about the jejunum, so in jejunum the carbohydrates got digest, nucleic acid got digest, fat got digest. Okay, for example, that which means that jejunum basically convert the disaccharide molecules into the monosaccharide. Okay, monosaccharide. If we talk about ileum, so ileum is having a absorption. Ileum basically works for the absorption, where the absorption of the molecules monosaccharide basically occur. Okay, so if we talk about ileum, so ileum is basically 3.5 meter. Okay, and here basically the absorption will occur because of the more villi, because if you talk about the small intestine, so in small intestine the more villi are in the ileum. Okay, so here the basically vitamin B12 will get absorbed, our got minerals get absorbed, electrolytes. Okay, fat, which was lipoprotein, as well as the nutrients got absorbed. Okay, and if we talk about the reabsorption, so reabsorption will occur in the large intestine. So we are having next topic, we are having a large intestine. So okay, let me give you the overview. So in large intestine, the reabsorption will occur of the salts, water. Okay, so this was about the overview of the small intestine, which we have discussed about. So this was the topic, that was the absorption of the nutrients. So in the next video we will discuss about the large intestine.